Good day and welcome everyone. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. It's my privilege and honor today to welcome our speaker, Dr. Satrajit Ghosh from the McGovern Institute for Brain Research at Massachusetts Institute of Technology. Dr. Ghosh received his Bachelor of Science degree with honors in computer science from the National University of Singapore. He then earned a PhD in cognitive and neural systems from Boston University. Dr. Ghosh is principal research scientist at the McGovern Institute for Brain Research and an assistant professor of otolaryngology at Harvard University School of Medicine. He directs the Senseable Intelligence Group, whose research portfolio comprises projects on spoken communication, brain imaging, and informatics to address the gaps in scientific knowledge in three different areas: the neural basis and translational applications of human spoken communication, machine learning approaches to precision psychiatry and medicine, and preserving information for reproducible research. He is also the director of data models and integration as part of the ReproNIM project and NIH P41 Center for Reproducible Neuroimaging Computation. Finally, amongst his many accomplishments, Dr. Ghosh is the co-PI of the DANDY project, a US Brain Initiative data archiving effort for cellular neurophysiology, as well as one of the prime movers of the NIPIPE open software development effort. Dr. Ghosh's lecture today is entitled The Shifting Dunes of Data and Computation. And as always, we are streaming this lecture live and for recording via YouTube. If you are watching via YouTube today, thank you so much for joining us. And also, if you are one of our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants, you are encouraged to submit your questions for Dr. Ghosh via the chat feature in your Zoom session. I will synthesize these questions and I'll ask them on your behalf towards the end of the lecture. And with that, welcome, Satra. Thank you so much for joining us. We're really looking forward to your lecture.
Hey, thank you, Jack. It's a pleasure to be here. And before I move forward, I will want to acknowledge the efforts of so many collaborators and members of the group over the many years. And I'm going to kind of span a couple of decades of information today. So I want to thank all the people who have contributed and influenced many of the things I have done and we continue to do today. We're also very grateful for support from the National Institute of Health, support that we've received from the INCF over the years, and also internal support from the McGovern Institute Neurotechnology Fund. As Jack mentioned, our group kind of focuses on three broad areas or branches of study. We want to understand the neurophysiology of human communication. We want to do applied machine learning, especially in the context of precision medicine and psychiatry. And we want to preserve knowledge through reproducible research and other neuroinformatics approaches. But what I'm going to focus on broadly today is in this brain imaging and analytics section of our work. And I wanted to start by simply showing what we can do with a single subject today. This is not super complicated, but we can look at things at an individual level on a brain. And what you're looking at on the left is an inflated brain. It's like taking the brain and blowing it up a little bit like a balloon so that you can peer into the cracks and crevices or the gyri and sulci of the brain. And on the right is the stimulus that the subject was seeing. And we're going to look at a real-time functional activation as the person is looking at those stimuli. I should note that this video has been edited to compress it into a very short time span, but it does follow the patterns that you would expect to see. I wish I would play music for you at this point. And at this point in time, the person is reading this aloud in the scanner. And as you may have noticed, there were some very specific patterns of activity as they were watching these different components of the tasks. But we did not quite start in the space where we could do this easily. We had questions around speech production. And scientific questions often need specific data and tools. So when we started, we were looking to build a model or a neurocomputational model of speech production. And we were looking at how neuroimaging could inform us to build such a model. And one of the papers we were looking at that point in time was something about the computational side of neuroimaging. This is from Acknowledgements, who was looking at a standard process at that time, which was to take brains and normalize or register them to each other. And when people did that, what Acknowledgements did was looked at Brodmann areas 17 and 18, parts of visual cortex, and said, when you align people using this normalization method, as you increase the number of overlapping brains, the number of voxels or pieces of the brain that actually overlap across people starts diminishing rather rapidly. Now, this was important for us in the context of us trying to study the speech motor control system. And so we took that kind of effort onto the temporal lobe and asked the same kinds of questions and demonstrated the same kinds of things. And this led us to the notion that we couldn't be using the standard tools in use at that time to try and understand how the speech motor control system worked. So painstakingly, every single individual was anatomically parcellated into regions of interest that then we could relate from one individual to another and study to understand the functional architecture of speech motor control in the brain. So on one hand, we were doing the science of trying to understand speech motor control. On the other hand, this required us developing probably our first set of tools. We created a MATLAB-based interactive editor that Jason Torvill, and for those of you who do brain imaging, the T in Torvill is the same as the DKT in the DKT Atlas that's distributed through FreeSurfer. And Alfonso Nieto Castanon, who today you might know as the lead developer of the CON library. At that time, we created this MATLAB interface for Jason to quickly parcellate, and quickly meant several hours of work. On the right, you kind of see an operational flowchart. And this workflow seems rather straightforward and simple. You get scanner data, you analyze it, you take the pre-processed data, and you bifurcate that into a couple of different analyses. And this entire thing created what we would call perhaps the first batch processing system at that time for SPM. SPM's batch editor had not yet been built, but it also was a place and time when we were developing things in MATLAB, writing code in C and C++, interacting with VTK and FreeSurfer, and putting computational geometry into our tools. And it gave us a foray into creating a set of tools that helps one answer specific questions when standard tools are not possible. It was also around that time that you could get data, but get data by having it mailed to you. And this is an example of us requesting data from the fMRI data center, and it was mailed to us in CDs that we could then load and take a look at. The world started changing about 4 or 5 years later. And one of the things that started happening was an explosion of the set of tools, packages, and algorithms that were available for neuroimaging. We saw an exponential growth in these packages, but they were also containing differences in algorithms. They made differences in assumptions. They ran on different platforms. They had different interfaces. They often relied on different data formats for some of these tools. So as the space was growing, the world of neuroimaging was starting to ask the question, could I use different packages for different kinds of things? And here's an example on the right of a section in a paper around fMRI data pre-processing. And you're already starting to see that people are using a couple of different packages to build together workflows and pipelines to process such data. It was also around this time that data started getting much more multimodal. We were acquiring anatomical images, we were acquiring functional images, we were looking at diffusion images for looking at water diffusion as a proxy for connectivity in the brain. Around the same time, the ecosystem around Python was growing. For those of you who might consider Python a de facto thing today, around that time when we started building the NiPy project, we had to put up this slide on Introduction to Computer Science and Programming at MIT being taught in Python to convince many people that Python is a real language that people should be using to do scientific work. And there was an incredible community of people led by many people around the world developing these scientific tools in Python. And this is what led us to one of the first tools we created that was distributed widely called NiPy. And the idea behind NiPy was simple. You had all these different packages. What we wanted to do was to collate those packages under a single uniform Python API so that the user or the developer of the workflow software did not have to learn how to call SPM through MATLAB or how to call a command line interface from FSL or FreeSurfer. It all came as a uniform standardized API that they could use. This did not mean that you had the luxury of not knowing what those tools do, but it just helped kind of putting these tools together to construct workflows that allowed you to mix and match things, especially as you're starting to get into multimodal data from anatomy, from functional data, and diffusion data. You could also use this engine to distribute your computation. Initially, it was to workstations in the lab at that time through SSH. And later on, it became one of those things that you could run on HPC clusters. And Chris Gargolewski was one of the lead architects of making this happen. He was in Boston for 6 months. We released one of the major versions, first versions of NiPy over a 1-week sprint that then I think changed the landscape of some of the pipeline-based computing that we see today. NiPy is in use in almost 88 countries today, and it has spurred the development of several packages that rely on it and an entire new ecosystem of tools and workflows that were spearheaded very much by Chris and Oscar and the other Chris, who's now helping maintain many of these different pieces. So this ecosystem is what we see today, but it started about 10 years back. And it is at that point in time, 10 years back, that we were looking at various ways of accessing data, software openly, also creating collaborations that were open. Some of the online systems like GitHub had just come online, and we were able to leverage some of these things to accelerate the software or the analytic collaborations that were happening. And it was also spearheaded by 2 other things. Around that time, the International Neuroinformatics Coordinating Facility, or INCF, created the Neuroimaging Data Sharing Task Force. And the Neuro Bureau started collaborating on creating an open space for scientists and researchers to work together collaboratively through brain hacks and other events. What was important at that time, and this was, again, looking at about 10 years, 9 to 10 years back, that we were already starting to think about how to capture information in a way that is reusable. A few weeks back, I know Marianne Martone gave a talk in this seminar series on fairness, or fair data and available digital objects. Some of that started at this point in time. We were looking at how do we represent metadata? How do we develop methods to ensure interoperability across different tools and packages? And how do we distribute this in a way that it's reusable by others? And this started a project called the Neuroimaging Data Model Project, which involves many people over the years in trying to structure information that's accessible at various levels. We built this specifically relying on something called the Provenance Model, something that would allow us to increase our trust in our data and results by knowing where they came from. The core technologies behind this were one of the things that was written in this CERN document almost 21 years back at that time by Tim Berners-Lee, in showing how one could relate different pieces of information on the web. And this is the model that we are using even today. And it is increasingly becoming clear that a lot of information that's coming online needs to be connected and related to each other. Provenance needs to be tracked. So the neuroimaging data model was one of the starting pieces of this. A key piece of this was also about harmonization of information. At that point in time, we were starting to get access to thousands of brain MRI images through initiatives that were spearheaded by Mike Milham at the McGovern Institute and Cameron Craddock. We got access to a lot of data. This was made available through an interactive interface where one could search for data sets through different aspects of phenotypic information. And it could be things like diagnosis. This involved individuals with ADHD or autism or control subjects, or they could be looking at various other aspects of phenotypes. And this created what we at that time called the Open Neuroimaging Laboratory, and perhaps the earliest use of the word open neuro, although open neuro today is a very different thing. It's a fantastic resource of data created by Chris and Russ and others in distributing fMRI data sets. The intent of creating a distribution like MetaSearch to be able to search through it was not just accessing data or seeing what's out there publicly. It was also about creating a community of people who would come together to edit brain images collaboratively using a browser-based platform. And this was one of the first forays into browser-based technologies for collaborative work. And this is something that's continued on today by Roberto and Katya. And one could go in there, collaboratively edit a brain, and then render a brain, which in this particular case is that of a black rhinoceros. But a lot of it was intended as going across brains, not just across species, but a lot of human brains from different projects. So this led to a lot of data coming online, but the data came online in different ways. And one of the things that happened with NIDM was, as a graph and a semantic web model, it was partly seen as fairly complex with no tools and technologies to actually create the metadata or the needs that would be, or the descriptors that would be required to express data in searchable form. On the other hand, you had completely unstructured PDFs, which people are still used to today. You read through it, you understand what was distributed, and then you go find it. The brain imaging data structure, which was formulated around this time in 2015, chose this middle ground of coming to a community consensus about a file layout and organization with consensus metadata. It did not try to make it rich with the linked data semantics that NIDM does, but it provided something that was acceptable to a large community and a set of technologies that the community could understand and build towards. And I think it's a fantastic example of how you want to think about tools in the context of your audience, as opposed to perhaps the complete notion of technology developers that might be needed to build an infrastructure around it. So I'll take the Human Connectome project as an example. While here I'm saying it was primarily unstructured data that you had to find and do things with, it ushered in a set of data that was fantastic. Today, that project has grown and will be releasing even more data over the next year. But when it first started, the primary data set that was available was the Young Adult Human Connectome project. And in that data set, which at that point we were playing with, had about 500 subjects. So that was already a few orders of magnitude larger than most data sets in people's labs. And this raised various kinds of challenges for us. One of the things we were interested in was to try and understand whether resting state functional data could be used to do better alignment of individuals to each other. Remember, very early on, I said one of the things people normally did was to take volumetric images and align one brain to another or to a template. We found that to be difficult, at least in those early days, and used region-based analysis to do things. But anatomical regions, while they cover some aspects of functionality, are not an identical map for all aspects of functionality at the macro scale level. So resting state data, which literally involves somebody lying in a scanner and you're monitoring their brain activity as it spontaneously fluctuates over time, provides some amount of structure to the patterns that emerge. And this gives rise to specific types of networks. And our question was, could we use this to look at how two people are similar to each other? And we relied on some mathematical techniques, which creates this matrix of correspondences. Now, the Human Connectome project data allows us to do this for every single individual. Here's an example of looking at it for 3 individuals. They may have different regions in the brain that relate similarly to each other, but you can kind of align them all together using a Procrustes or other types of transforms into a common space. Now, the challenge here was that the Human Connectome project data released data that were about 90,000 rows or points. And if you construct this matrix, that would be a 90,000 square matrix. If you stored the 90,000 square matrix on a computer, it will, as a floating point number or a matrix of floating point numbers, that will take about 64 gigabytes of RAM. At that point in time, not every computer had 64 gigabytes of RAM. And if you couple this now with 500 subjects worth of data, you're getting into a place where your standard computational tools are no longer feasible to do some of these things. So we came up with various ways and efficiencies to deal with that scale of data, and that changed the mindset of what computational infrastructure we should be investing in, even at MIT. We were able to, at that time, think about ways in which the future of computation were to evolve and preempt the construction of our compute cluster to leverage larger memory sizes, even though most people weren't really solving problems that needed that much. And this was a big boost or a benefit at that time. We were able to run this Human Connectome project analysis where we aligned people to a common space using functional architectures. And this itself led to a series of new ways of applying this kind of embedding or alignment that was spearheaded by a dear friend and colleague, Daniel Margolis, in looking at gradients in the brain, both within and across species, as a way of representing macro scale gradients of information that go from unimodal properties to heteromodal regions that have now allowed people to look at this in the context of various disorders, looking at within region gradients, and kind of thinking about how these gradients map onto different aspects of behavior and cognition. Well, many samples were kind of useful. One of the things that we were still interested in was going back to our understanding of human communication physiology. So we wanted to see how a standard diagram in a textbook, like the one you see on the left, maps on to actual human data. Now, it turns out that for most part, these diagrams are based on animal or other studies. And just looking at the auditory and visual systems, one might notice that one of the primary differences are the different stages of brain regions or layers that one goes through, going from the sensory organ, so in the auditory system, the cochlea, or the visual system, the retina, into perhaps the first major thoroughfare in relation to cortex, the thalamus. And the auditory system has many more junctions, whereas in the visual system, that has been compressed and a lot of computation gets done in the retina itself. We were interested in trying to see if we can figure out this pathway in vivo in humans using these technologies that we had access to, like MRI. Turns out we couldn't quite get there. So instead, we stepped back. We still dealt with the human system, but using ex vivo tissues. And we were delighted to find a data set that was available that involved MR imaging of an ex vivo brainstem that goes all the way from the brainstem up to thalamus. And these are some of the images that come out from this data set. So on the top row is this ex vivo data set. On the bottom row is kind of a standard human connectome project 3 Tesla scan of the corresponding anatomy. And as you might be able to see in this display, the incredible richness of the data that comes from the ex vivo scans. So we were happy to see that many people were starting to put data out there that they weren't intending to be used for the purposes we use them for, but by putting the data openly out there, we were able to reuse it for a project that it wasn't originally necessarily created for. And here is looking at the diffusion orientation distribution functions. And again, a comparison on the left of the ex vivo data and on the right of the human connectome project data. This is truly an unfair comparison. On the right is an in vivo scan. On the left is an ex vivo scan. But I think it's very clear the structure and the clarity that comes just from looking at these orientation distribution functions on the left relative to the in vivo scan, where we still have some ways to go to get to the kind of clarity that the ex vivo scan provides. And this allowed Kevin, who was a grad student in the group at that time, to create a replica of that textbook figure using the diffusion data that we got from the ex vivo scan. Now, for those of you who do diffusion MR tractography, there's a lot of noise in the signal. So it's a little difficult to necessarily trust everything you see or get out of a diffusion tractography reconstruction. So as a following step, another grad student, and this is Federico Di Martino, who's at Maastricht University in the Netherlands, his grad student, Farooq Gulban, kind of joined up with Kevin to say, what can we do to kind of extend this even further? And what they did was leverage a couple of public data sets, the ex vivo data set that we were looking at, the big brain data set, which was released from McGill and Jülich, and in vivo MRI data at 7T that Farooq and Federico had collected at Maastricht, in creating a connection between the postmortem MRI, the big brain, and the in vivo MRI. And this allowed us to look at test-retest reliability of the localization of these functions across individuals and look at different structures across test-retest within an individual on different experimental days. Now, this kind of work relied on many different new techniques to be created, including alignment of brain data across in vivo and ex vivo scans. It turned out when we initially did this work, the registration methods failed, and Farooq and Kevin had to go in and tweak those registration methods to make this work. You might also notice these were at very different resolutions or scales. So most techniques were created not to address that large discrepancies in resolution and were relying on resolution being somewhat similar to be able to align things to each other. So this also required a new set of tools and techniques to be generated and looked at much more prospectively and closely to see if these registrations worked for people. But this was one of the things that this produced. And in addition, whatever was generated as a result of this was made publicly available as open data and other compute resources. Shifting gears a little bit in our group, the other thing that we started looking at around that time was looking at mapping genetic information through brain imaging data into some level of clinical information. And in this particular case, we were looking at schizophrenia with collaborators Kihan Batmanglek, who's now at University of Pittsburgh, and Joel Sanchez Araujo, who's now a grad student at Princeton. And we were interested in seeing if by inserting an endophenotype, so instead of going directly from genetic data to the clinical state, if we inserted an endophenotype like brain imaging information, could we get an easier process to both learn the mapping from genes to the diagnosis state, or also map brain regions back to genetic precursors? And we tested this by asking the question, how well could we predict the state of the individual using these two different methods? So in one case, we just went with a standard mechanism like lasso or other machine learning techniques that went from brain regions to the classification stage. In the other case, we used a probabilistic graphical model to map the genes through the endophenotype to the disease state. And what we found was that the probabilistic graphical model had very similar performance to the standard machine learning technique. The difference being it was much, much sparser just by the nature of the construction of the model. We also ran an independent validation on some external data sets, and that also showed us that this model was doing pretty similarly on data that it had never seen. And it provides back a marker back to the genetic information. Now, what was interesting at this time was this was one of the earliest data sets that was available through the Genus Consortium, thanks to Gabriella and Tracy, that had a decent number of subjects with brain imaging data and genetic information. We were able to constrain the genetic information through other much larger scale studies like the PGC study, and we're able to include different kinds of phenotypic information to further regress out variance in this sample. But this led us to the fact that over the years, we saw this increasing trend in multimodal data sets coming into play, which requires new analytics, new tools, and new ways of looking at and exploring the data. So what does the next generation data and compute infrastructure look like? One of the things that people have access to now is a fairly large set of data. You have various biobank projects like the UK Biobank, the Genus project that I just mentioned, the ABCD study that's collecting longitudinal data across about 10,000 kids, the Healthy Brain Network study that's also plans to track 10,000 kids, and at this point in time is around 3,600 samples or so. The FCP Indy project, this is the functional connectome project, the Open Neuro, and the ongoing human connectome projects and its extensions that are all giving rise to a scale of data that's very different from what people are used to in their labs. And these data, excuse me, are spanning multiple modalities and allows for rich exploration and investigation. And this requires a slightly different way of approaching things. And for this reason, we just finished running the first iteration of the ABCD Repronym course, which was targeted at mixing computational tools with exploring the ABCD data. A similar course has been run by Ariel Rokem and Tal Yarkoni for the last 4 years called Neuro Academy, which again focuses on these computational tools that will be necessary to process these scales of diverse and heterogeneous data and large-scale data that's coming online today. And then over the pandemic, one of the fantastic things that happened was NeuroMatch Academy, which was an online school for computational neuroscience, trying to integrate computational neuroscience techniques as well as giving students an option to learn about the various tools and resources that one has access to. And some of these are being coordinated through a platform called Neurostars, which is an open platform for questions and answers for the neuroscience community run by the INCF. And I encourage everybody to contribute and build this platform in looking at and responding to questions that people create. Alongside these large data sets, the next problem that kind of comes up is, how do I access this data? And on that front, one of the projects that has been coming online over the last 5 years or so is a project called Datalab. And what Datalab does is it gives you access to a plethora of data across the planet through a Git-like interface. Now, Git may be something that you're unfamiliar with. It's a version control system. And what Datalab does is it turns that to version control data and provide access to information that otherwise would have been difficult. As is shown on the slide, the amount of data that you can access through Datalab is about 268 terabytes today. And this is growing at an exponential rate. Most people probably don't have space to download that scale of data onto their HPC systems or clusters. And this is what the unique part about Datalab is. You don't need to download 268 terabytes of data. You can download only the necessary bits that you need access to. So in some ways, it provides an index to an incredible assortment of data and allows you to search for and get to pieces of data that you need for the question that you have at hand. And in case you have a few petabytes of storage, you could download all the pieces of data and store it locally. Alongside this access to data, we are seeing an incredible set of tools coming online. Some of these are as old as that first slide I showed. SPM, FSL, FreeSurfer, ANS, and AFNI have been around for a long time and continue to be some of the key elements that are used in neuroimaging analysis. But there are also a whole lot of tools coming online in the machine learning space, including NiLearn, which is built on top of scikit-learn, and the various kind of standard machine learning modules of TensorFlow, PyTorch, and Keras. On the neuroimaging side itself, we are changing the ecosystem a little bit, building new general purpose tools like Pydra as a new workflow engine, TestKraken, a new testing framework, NeuroDocker, creating analysis environments for Docker and Singularity, and NoBrainer, which I will talk about in a little bit of time, a brain DNN library. And alongside these are highly specialized other tools like Huricon for DICOM conversion, MRI-QC, various of the NiPreps and NiFlows that are coming online. There are also specialized interfaces being built for analyzing certain types of data. In this case, analyzing data from fMRI studies where people were watching movies. And this is a project spearheaded by Talia Arconi and Alejandro de la Vega at University of Texas, Austin. And it's building an online platform that allows you to quickly select the kinds of questions that you might want to ask of these data sets and create an analysis module that you can then run on your own cluster to get at results from those questions. Alongside such a specialized interface, there are other ways of looking at literature. And from Gael Varroqua's lab, one of the projects is NeuroQuery, which allows you to ask questions almost as if you were asking someone a question. And it will take the entities or patterns in the question or the query and turn them into a brain map indicating places or regions in the brain which might have been responsive to those terms or entities that were in the question. And it will link that brain map or the creation of that brain map to the set of publications that were used to create that brain map. So allowing a rich connection between a query and literature through brain imaging coordinates and data. So this brings me to, I mentioned NoBrainer earlier as a robust and validated neural network tool suite, and this is a recent Brain Initiative project that we are working on with a bunch of collaborators, but primarily it's being run through MIT, Mass General Hospital, and Georgia State University, with some collaborations at NIMH and outside of the States. The current state of affairs is, as you have just heard, an explosion of public accessible data. There's all kinds of data coming online. There's also been an explosion of models. You have models being created for segmentation, behavior prediction, connectomics, diagnosis, and we're seeing a continued improvement in the computational infrastructure that supports these models. However, there are many challenges. Despite all the data being available, I would say that it's still the tip of the imaging data iceberg. There's much more clinical data that's hidden in hospitals. Also, much of the open data have been anonymized. So they have gone through transformations which make it easy to access data, but are not realistic enough necessarily in relation to data that you might get straight off of a scanner. Many of the models that are being distributed, I would say, still lack a fair amount of fairness. While code is being made increasingly available, the pre-trained models are not. Now, this is all changing dynamically even right now. You are getting more models that come out. But we're also seeing a rush to creating models which don't necessarily have robustness or validated across a very large set of data sets. And often these are research products that are not engineered for production or computational efficiency. So if you wanted to run some of these models on the size and scale of some of the biobank data, they would take time and effort to do so. There's also limited consideration of data bias in many cases. And finally, a lot of underspecification is happening as we build some of these models, which limits the utility of the models beyond the lab into real world spaces. So we have been building up this framework called No-Brainer to try and kind of address some of these challenges. And here's a couple of examples that I would like to share with you. We've been building some base networks for image segmentations and parcellation. We'll move on to time series processing and surface-based operations pretty soon. We were able to show that a brain extraction network trained on 10,000 scans could be quickly fine-tuned to provide an improvement for meningioma segmentation by relying on the initial network trained on the 10,000 scans, as opposed to training just on the meningioma segmentation data. Together with colleagues at NIMH, we've developed this Bayesian model or network to not only segment brains, but also to give us a measure of uncertainty, which allows us to relate information in a brain to the quality of the image itself. And this has allowed us to kind of think about quality control or how to improve the quality of public data sets right from the get-go, instead of every lab having to think and reevaluate quality whenever they download the data. But this comes at a little bit of a cost. In developing this project, you need to understand various aspects of infrastructure. It is a Python library. That part is straightforward. But we are also using Datalab and open science framework to distribute models and data, and creating containerized applications that one would need to understand how to use containers to run and use in their own centers. And when we build these resources, we have access to a set of resources here at MIT that spans different kinds of hardware. And if somebody had to come into the project, they would need to have some kind of background to be able to deal with these different architectures, infrastructures, and platforms, and be able to understand the data modalities and infrastructure that deals with creating such models using brain imaging data. And this is a challenge for us right now. Getting a set of people who have the knowledge that spans these domains is hard. And it's something that would have to change at a fundamental level in how we think about training individuals computationally at universities and other places. In neuroscience, one of the things we want to be able to do is to move beyond a single modality and get towards multi-scale and multimodal integration. And this is changing very rapidly here, at least in the US, and more broadly across brain initiatives in the world. The US Brain Initiative has funded and supported a large set of data archives to capture data from subcellular resolution to the whole brain scale. And one of the projects we are lucky to be part of is called DANDY, which we're treating as not just a data archive, but also hoping that it will change the way people interact with data and with each other in the research space. So we call it a collaboration space for cellular neurophysiology projects. The basic elements of DANDY, like any other application, is fairly straightforward. It's a web application that you can use to browse various data sets, but it uses standardized descriptors, which improves the fairness of data being distributed through DANDY, We're relying on things like Neurodata Without Borders, BIDS, and NIDM as standards that inform the data coming into the archive, which makes it easier for people to reuse some of these things when they download the data onto their own platforms. Alongside the web applications, we are running a Jupyter Hub, which is an analytic platform, providing the ability for people to do computation very close to the data on the hub. Over the last year, we've grown from about 2 terabytes to 13 terabytes of data. And this is currently on a little bit of an exponential trajectory. So I hope we would be able to kind of think about as a community what kind of data we should be preserving in the long term. We don't have an answer for that right now. Now, to reuse this data, the data has to be richly annotated with metadata, and we are working with various groups to try and do so. And people who are using this archive can use various Python clients to get at data into and out of the archive. Now, the reason we were able to build this up fairly quickly was we were relying on a set of open source technologies with a view that many different kinds of people could come and use data from this archive. It could be a hardware developer who says, I want to make it easy for people to submit data to Dandy, and I'm going to put out data in a common format. Or it could be a citizen scientist, a high school student, or someone who comes and says, I want to learn more about neuroscience data, and this data is now accessible at their fingertips on the archive. And we have leveraged a lot of key projects that have been built by the open source and open science community over the years that we have been able to kind of click together or patch together to create this archive. And at this moment, it has data from 4 species. It supports multimodal data from intra and extracellular recordings, optical data, MRI, optical coherence tomography data, microscopy data, immunostaining data, and human ECOG data. It can support multi-session data, and it supports various kinds of behavioral, both manipulations and observations through optogenetic, pre-behaving, or video data sets. As I said, one of the key elements of any data distribution today is getting information in, and this still remains a challenge for us. We want information that comes from the scientist or the researcher to make the data more useful to the consumer or the end user. And the general tagline I go with is, if your lab burned down, would you have put enough metadata onto the archive so that you can restart your lab with the data that's living on the archive? And this requires a concentrated effort, not just within a lab, but across a community to come out with better standards for describing the data. The amount of metadata that's needed or necessary to interpret the data is absolutely going to be critical as we put these archives online. The other challenge that's happening is the amount of data that's coming online. NeuroPixels is a probe that's inserted that has about 960 channels over 10 millimeters. You can insert it at multiple regions in the brain, collect data. And about a 13-hour recording of data would be about a terabyte in space. Now, if you imagine and scale this up across labs doing such studies, the amount of raw data being generated is going to soon explode, and we will have to come to think about how to preserve data and what to preserve. The data generation and processing is increasing on a daily basis, and you might be looking at 10 terabytes per day of data across labs, all the way to microscopy, where you might be looking at hundreds of petabytes of data over time. This requires not just challenges to be addressed in terms of standardization and descriptor evolution, but primarily around sustainability. How are we going to sustain that scale of data? Transfer and storage are going to be equally important things, both locally and in the cloud or across networks. And how do you compute on such data? We are already saying that computing on some of the data sets might need to happen very close to the data, as opposed to pulling the data from wherever it is through a mechanism like Datalab and computing on it locally. And finally, to be able to truly extract knowledge, we need data sets to be interoperable, in the sense that they have to have sufficient metadata to be able to be reused for other purposes. At this point, I think the culture of sharing has evolved quite well, but we are still expecting a lot of evolution in the process of sharing data as soon as it's generated, collaborating with others in the cloud or on data centers, and then creating a community of practice around the best practices that would be required to distribute data, to compute on it, and to evolve and coalesce into well-validated workflows that might exist. And this kind of leads me to the last section of this talk, a little bit of forward looking as to what we might expect. And I'll go back to the work we do in our group. Some of the things we are interested in is the ability to measure things about an individual, to predict outcome like treatment or behavioral trajectories, and to adjust how an individual might behave through interventions or the way they do things, and be able to remeasure. And this is kind of a cycle. What we are interested in is thinking about trajectories across individuals that might overlap, diverge, or change to certain states that might cause illnesses. But to be able to do this, we need both objective and subjective measures. Now, objective measures are growing on a daily basis with various kinds of wearables and other data sets that are coming online. But this is really about longitudinal data about an individual. And we are hoping that we can feed this into some of our models and algorithms that learn from the data to provide better or more precise indicators to an individual or their caregivers. And this will require what many people have said as deep longitudinal phenotyping. In fact, this figure on the right is from about 5 years back. NetApp, a company that specializes in storage, was already thinking about how the human body would give rise to a large amount of data as time progresses. And if you now think of this as kind of longitudinal data, every single human is going to be producing scales of data that you might not have been thinking about previously. And this might be brain images and signals, but it's also data from smartphones and wearables. The genetic data, especially if you now consider transcriptomic and other types of genetic transformations, are exploding at a rapid scale and size. Clinical records are more accessible, although there's lots of difficulties in getting to them right now. And then the world of microbiomes is also changing some of these scales of data as we speak. So how we preserve even our own individual data is something we have to think about moving forward, not just institutional and structural information that's stored in clinical centers. And one of the projects I really appreciate in this space is a project called Open Humans, which allows storing your own data into a personal data store. And what this means is that you have control over your data. You decide who to give it to. You could donate it for research, you could do your own research, or you could explore things by yourself. And I think it's a fantastic model for us to consider as we move forward in thinking about both privacy considerations, but also being able to aggregate large amounts of data when we need to solve problems. And this will still require the kind of work that we were doing in the neuroimaging data model, but the scale of all biomedical data. We need to be able to link, extract, search over information, even if the raw underlying data may not be accessible. We may need to compute over information in privacy-preserving ways as well. So it's something to keep in mind as we think about the scale of data. If we could do this, maybe tomorrow, well, maybe not tomorrow, but maybe over a short period of time, we're able to ask questions for specific disorders where there may not be large amounts of data in these public archives. We might be able to develop algorithms and tools that actually operate on real data and takes care of bias across data sets. And this will require access to data in a way. And even Open Humans, where the model is fine, would still require evolving to the scale and tune of data that's coming online. And one of the projects in that space is called the Interplanetary File System. It's a way of distributing data across nodes and networks in a way where data is not just stored in a central archive or a central repository, but gets distributed to make certain aspects of data access and distribution much more efficient. Now, this may not be a solution for all biomedical data tomorrow, but this is an architecture that one can think about in thinking about both private data sets, public data sets, and distributing data sets over the planet. The final thing I'm going to end with is the need for education. So on the left, the missing semester of your CS education is a website at MIT that teaches people about the things that are not often taught in courses. And these are critical in the world of analytics and data systems that exist today. Some of the things that every student should or researcher should know how to be able to do is to make their computation portable, to be able to go from one system to another, to be able to transition from one hardware architecture to another. There has to be a better way of doing debugging. The iterative process that's often involved in many of the analytic tools, especially deep learning and other technologies, requires us moving or iterating on the problems faster. We have to be able to track provenance if we have to show people what a model was trained on, how well it can do. And metadata curation, which is something that's often ignored in most analytics places, needs to become a more concerted effort and a systematic effort across researchers and labs. We have to be able to compute efficiently at the scales of data we are seeing. Otherwise, we will be waiting for just computation to finish. Bias and quality control are going to be essential with the rate at which data are emerging. A lot of data are being put out there without quality control, and that's a good thing in my opinion. However, as a community, we need to be able to label or annotate the data sets so that downstream users can decide whether they want to use certain types of data. For all of the software and analytic tools, testing and reproducibility and validation are going to become critical. And for many domains where we don't have ground truth available, the community kind of has to come together to establish best practices or principles by which we evaluate, not just from looking at the result and whether it aligns with our hypothesis, but some more principled mechanisms, whether through computational modeling or others, to say that what we are observing as a result of the analytics is actually what we want to see. And the scale of data is going to parallel the scale of computing, which will go back to the kinds of tools and technologies that a student or a researcher might need to learn today to be able to deal with this explosion of data that's happening. With that, I'm going to thank you for your attention, and I'm happy to take any questions.
Satra, thank you so much, man. You covered a ton of ground. That is really fantastic. Let me see, couple of questions. We have one from one of our viewers here. How do you quantify or mitigate uncertainty when deploying machine learning techniques in your data analytic quests? Are there confidence intervals? Are those a big deal? Should we be paying attention to those?
Wonderful question. So I can say about one of the approaches we are taking with Bayesian neural networks, which as an inbuilt mechanism of that architecture provides us access to entropy or uncertainty that we can leverage. So when it runs, it kind of says, this is how certain I'm about what I'm telling you. Now, many of these things, as one might know, could be confident about some things, and we haven't solved every problem with uncertainty, but at least using these Bayesian neural networks is one step of evaluating uncertainty that we think can be useful in the short term.
One thing that is super interesting about kind of the story you just told is, kind of beginning with an effort I was involved with moons ago, where we were delivering data to people on DVD. And if you kind of think about that time, it was about the same time that like Netflix started to become popular. And how did you get your movie of the week from Netflix? Well, you ordered it and they delivered it on a media like compact disc or DVD. And so it wasn't unusual. And we were very sensitive to the fact that, filling up somebody's Ethernet pipe with, streaming neuroimaging data would very often bring some networks to their knees. And so we actually specifically chose that as a medium to act as kind of a governor on the system, right? So that we didn't have, a lot of people complaining at us that we, had basically having the investigators being complained about that they crashed their own internets about it. At the same time, realizing that the ability to stream effectively that kind of data writ large was going to be a real challenge. And we just, at the technology of the time, wasn't equipped for it. Even Netflix wasn't equipped. Now we take that for granted, right? We stream stuff to our phones, sitting on a bus or something. And so the technologies continue to move forward. And that is certainly something that we've seen in neuroimaging with all the different approaches and methodologies and services that you mentioned. And things like this, I hadn't heard of the IPFS, but you could imagine being able to do that. And I'm also was intrigued by the Net app, talking about the Net app thing of 665 terabytes worth of just individual data. That brought to mind the Quantified Self project, which was kind of popular a few years ago, but then linking into Open Humans. If we all quantified ourselves, that would be, and made that data distributable through IPFS and had all these quantitative tools, it would really be quite an impressive amount of stuff at our fingertips on demand when we wanted it. It would be quite impressive.
Absolutely. I thought you might get a kick out of that fMRIDC slide over there, Jack.
It was it's interesting. It was pioneering for its time, but I marvel at what we started because there's just been so much work done in this area, which is just so remarkable, and you just summarized it for us. So.
And one of the things I will say is we are not at the stage where everything can go over the internet yet. So I know of a project within the Brain Initiative Cell Census Network that sends data by tape drives because even the internet is insufficient to send that data over. So they're collecting fluorescence, micro-optical sectioning tomography or FMOS data, which are close to a petabyte if you collate all those tapes together. And that becomes almost impossible to send over the internet today.
Yeah, it's a real challenge, and we're going to have to think very carefully about how we, instead of moving data to where the processing happens, to possibly take processing to where the data live.
Right. And it has to be, I think, both of those things, which is why I'm excited about at least the future of things like IPFS. And again, I think whether that exact technology is what's adopted, I don't know the answer. But I think the concepts in that technology are really good. And we will see this kind of mixed movement of either data or computation in the future world of research analytics. And if that can indeed be coupled with the personal data stores like Open Humans, it just simply opens up a scale and potential for human-oriented research that I think would transform almost everything we're doing around human research today.
Well, Satrajit Ghosh from the McGovern Institute of Brain Research at MIT, thank you so much for sharing your work with us and all these exciting efforts. So thankful to have you join us. And we will not be having a data science seminar series next Friday, first because of some other activities that we're doing for our 2020-2021 Innovation Lab participants, but we will be seeing you again soon here in the future. Once again, thank you, Satra. Everyone enjoy a great weekend.